philosophical question. Recording. All right, good. The somewhat philosophical question of what makes us move. What goes off in the brain at a given time that allows it to decide when to spur the body into motion? Well, we know that when we move, thousands of neurons in higher motor centers suddenly begin firing together in concert. And this strong coordinated neural activity is relayed via spinal cord motor neurons to muscle fibers that power movement. But this isn't satisfying because, of course, it doesn't explain why those neurons started firing together in the first place, which is really the crux of the issue. Now, one idea is that this activity could come from sensory areas. For example, if I throw this football to you, that strong activity charging through your visual pathways could be passed along to motor centers to trigger the coordinated activity needed to move. But not all movements result from overt sensory cues. For example, consider a cup of coffee sitting in front of you during this talk. Now, presumably, it's been sitting there long enough that it's no longer causing bursts of sensory activity in your cortex. Yet, mysteriously, at some moment, your hand may reach out, pick it up, and take a drink. Now, here, it's not so simple to imagine where this motor activity came from. I will operationally refer to these two classes of movements as reactions and self-initiated movements, whereas self-initiated movement is distinguished as one in which the brain has to rely on some internal process rather than an overt cue to generate it. It turns out it's not just intuition telling us there is something mysteriously different about these two types of movements. Clues from human movement disorders also suggest these movements are generated differently. In particular, we probably all know a little about Parkinson's disease, a movement disorder in which patients feel slowed down and have difficulty initiating movement. But what might surprise you is that patients particularly struggle to self-initiate movements like picking up a cup, yet they can react with relative ease to sensory cues like a flying football. This so-called paradoxical kinesia is so striking that assistive technologies exploit it. For example, there are walkers that display visual cues on the ground to prompt patients when to take steps. This striking difference in the way in which these two types of movement are affected in Parkinson's suggests that whatever is going wrong in this disease might be the critical clue in figuring out how the brain self-initiates its own movements. So what might be going on? Well, we've known for decades that there's profound loss of dopamine neurons in Parkinson's disease. These neurons live in a small strip of the brainstem called the substantia nigra, and you can actually see the neurons by eye from their dark pigment. This healthy midbrain has lots of dark dopamine neurons, but this Parkinsonian midbrain has lost its dark strip, showing us that these neurons are almost completely gone. The fact that these neurons are missing in Parkinson's suggests they must normally be vital to this process of self-initiating movement. But precisely how this degeneration gives rise to the symptoms of Parkinson's remains poorly understood, and it's been surprisingly difficult to ascribe a role for the healthy dopaminergic system to movement initiation. In fact, Josh Dudman's lab recently showed that when dopamine neurons are stimulated to fire at physiological rates, this fails to elicit movement, suggesting that whatever dopamine neurons are doing in generating self-initiated movement is more subtle than directly triggering it. But there's one other important observation from Parkinson's, and that's this mysterious slowing down. Of movement, certainly, but of cognition more generally. And although the literature is complex, it is undeniable that perturbations of the dopaminergic system, including Parkinson's disease, can alter both perceptual and behavioral timing in humans and in animals. We suspected that the Parkinsonian deficits in movement initiation and internal timing might both be related to one another via some shared dopaminergic mechanism. So recalling our original question, how does the brain coordinate its own motor system activity seemingly out of the blue? We asked what the motor and timing deficits of Parkinson's disease might imply about what dopamine neurons are doing to contribute to these processes. When we know that dopamine neurons that are lost in Parkinson's project densely to the dorsal striatum, where they exert a push-pull effect on the direct and indirect pathways. Now, this is a super simplified view of what these pathways do, but overall, we know they form parallel positive and negative feedback loops to the cortex, and one could imagine that these pathways could form a sort of cortical amplifier, with the direct pathway tending to enhance cortical activity, and the indirect pathway tending to keep this activity in check. In this view, higher motor centers may be able to self-initiate their own coordinated activity when the gain of this amplifier is just right. Or put differently, when the balance of the direct and indirect pathways tips in favor of the direct pathway. We hypothesized that dopaminergic activity could influence when movements initiate by tuning this balance, tending to push it toward the direct pathway, thereby pushing the system inexorably toward the moment of movement onset. To test this hypothesis, we recorded and manipulated dopaminergic activity in mice trained to make self-initiated movements. But to study self-initiated movement meaningfully, we needed a situation where the mouse had to rely on some internal process rather than an overt cue to decide when to move. To satisfy this constraint, we trained mice to do a self-timed movement task, in which animals received an audiovisual cue and then had to estimate elapsed time to determine when to initiate licking to obtain reward. It's vital to understand that the animal is not being cued when to lick, but instead has to estimate elapsed time to decide when to make her move. Now, because she has to rely on her own internal process of self-timing, these movements are implicitly in the family of what we operationally have defined as self-initiated. Now, mice, like humans, are pretty good at timing, but their time behavior is even more variable than ours. For instance, the mode of this animal's timing distribution is at 2.5 seconds, but sometimes he moves a second earlier, and other times he moves whole seconds later. And it's important to note that this behavioral variability isn't a bug. In fact, it's vital. It's exactly what we need to test our hypothesis, because with it, we can ask, what are dopamine neurons doing differently on trials when the animal decides to move early versus these trials when he decides to move late? To answer this question, we took a fiber photometry approach to record dopamine neurons in the substantia nigra, the population that's lost in Parkinson's. We specifically expressed the fluorescent calcium indicator GCAMP6F in these dopamine neurons using DAT-CRE mice and a CRE-dependent virus. GCAMP is a protein derived from GFP that has been modified so that its green fluorescence gets brighter when it binds calcium, and that allows us to monitor calcium entry into dopamine neurons. So when neurons are more actively signaling, intracellular calcium will increase, and so will the brightness of the fluorescence of this protein. And that's going to allow us to approximate the activity levels of neurons using little fiber optic telescopes that we implant in the brain. Now, this fluorescent calcium indicator diffuses throughout the cell body and axon, and this allows us to record dopaminergic population activity, both at cell bodies and at axon terminals, where these cells transmit their signals to the direct and indirect pathways. To do so, we simply implant a fiber optic over each of those brain areas. We also co-expressed a second calcium-insensitive fluorophore called TD tomato, which allows us to correct for any movement-related artifacts in our neural signal. So the mice completing our task were water deprived and head fixed, and they completed hundreds of trials of the timing task per session. On each trial, they receive a brief LED and tone cue to start their timing, after which the mouse has to decide when to lick in the absence of any further cues. The mouse's goal should be to make its first lick during the reward window, between 3.3 to 7 seconds after the cue, because if the first lick is too early or too late, it won't receive any juice reward for the trial. Now, since we're interested in signals related to the internal process of self-timing, we carefully monitored animals' body movements with 4 complementary techniques so that we could distinguish neural activity related to the timing from that could be more simply explained by ongoing body movements. So first, I want to show you the simplest possible picture of what we found when recording dopamine neurons. So this is the average of the GCAMP signal across all trials for one mouse where the response fell between 0.7 and 3.3 seconds. The x-axis is time, but there's actually 2 parts to this plot. On the left is a cue-triggered average, where all the signals have been aligned to the cue and averaged together, and this is to show responses related to the cue. But I also want to show responses related to the movement. But remember, movements are happening over a very broad range of times relative to the cue. So I split the x-axis here and plotted the average of all the trials aligned to the time of the movement on the right. Now the signals here are unfolding over the course of seconds. Let's focus on the clear things we see. First, we see there's a cue-related response. And then we see there's also a movement-related response. Now, neither of these features is too surprising. Dopamine neurons are known to fire transiently in response to salient cues and at the onset of movement. But what is surprising is the third thing we see, namely this apparent ramp between the time of the cue and the movement. This signal started several seconds before the movement occurred, and it appears as though dopaminergic activity is ramping up to the time of the movement. The rising of this ramp made us think of timers, so we asked whether the ramp was related to the mouse's natural variability in its timing. To test this idea, we pulled the GCAMP responses by the time of the movement relative to the cue. Now here we're looking at trials where the first lick occurred between 4 to 4.25 seconds, and we see the same rough patterns as before. Now, if we add in another movement time, we see this purple trace shows all the trials where the lick occurred between 1 to 1.25 seconds. And now we notice a few differences. The baselines start from a similar level, but begin to diverge even a little before the cue. The position at the cue onset is higher for the earlier trials. The ramp does not dip down as low on the early trials as it did in the later trials. And the early trials ramp is steeper than that of the later trials. Yet despite these differences, in both cases, the responses ramp to a similar level immediately before movement onset. It's important to remember that the differences between these 2 sets of pooled trials are the result of the animal's own natural variability in its timing. There's no difference in the task. Now, if we add in pooled trials that happen in between, We notice again that baselines start similar, but diverge before the cue, after which follows a clear fanning of responses that is already predictive by eye of when movement will occur, starting more than 4 seconds before the movement onset, which is a long time for neurons. So just think about it. Just by looking at dopaminergic activity, we can learn something about what the mouse is going to do seconds in the future. We saw this pattern in every mouse we tested. When we average responses across all 12 mice, we found this pattern becomes abundantly clear. So far, the signals we've observed and that I've shown you are related to the activity of dopamine neurons, but remember that we hypothesized that dopaminergic activity could influence the timing of movement initiation by tuning the balance of the direct and indirect pathways. In these plots, I've just shown GCAMP signals from cell bodies in substantia nigra, but I'll note that we see similar ramping GCAMP signals at axon terminals from these neurons in the striatum, as well as in dopamine release into the striatum, where dopamine influences the direct and indirect pathways. So, since dopamine neurons are communicating different signals to the direct and indirect pathways on trails of different movement times, we next asked what these signals might be doing in the brain. Now, since the differences in these signals seemed related to the mouse's timing, we wondered whether they could quantitatively predict the timing of movement initiation. So here to show more cleanly the pre-movement responses in the plot, I've truncated the average traces from our 12 GCAMP mice at the time of movement onset. Now, by eye, we see the dopaminergic signal is highly predictive of movement timing, starting even before the cue, both via baseline offset and activity, as well as dynamic ramping signals during the timing interval. By contrast, TD tomato control data didn't show any of these patterns, and GLM encoding models show that the ramps and baseline offsets were not explained by ongoing body movements. Now, because these signals looked reminiscent of the old pacemaker accumulator models of timing, we asked whether a simple threshold model could predict movement timing. In this model, the internal clock, or pacemaker, is assumed to collect noisy clock ticks starting at the beginning of evidence accumulation. As evidence accumulates, the signals follow a random walk toward a threshold, which, once crossed, appears to determine the animal's timing. We thus asked whether the simple threshold model for these dopaminergic signals could explain when an animal decided to move. And you can see by eye that we'll find that the time of these signals, when these signals cross this arbitrary threshold, strongly predict the movement time. And we know we could have drawn our threshold anywhere along the y-axis with a similar result. So given that dopaminergic dynamics were predictive of movement timing and were not explained by artifacts, we asked whether causally manipulating the activity of dopamine neurons could influence movement timing. To address this, we optogenetically activated or inhibited dopamine neurons on 30% of randomly interleaved trials. And because dopaminergic activity was higher at baseline and ramped up faster preceding early movements, we hypothesized that activation might cause more early movements on stimulated trials, whereas inhibition might cause more later movements. To test this idea, we compared the distributions of movement times on stimulated versus unstimulated trials. We found that dopaminergic manipulation bidirectionally tuned the timing distribution, with activation causing early shifting, inhibition causing late shifting, and no opsin control animals showing no consistent effect of stimulation, suggesting these changes in the timing distribution were not the result of the animals reacting to the stimulation. We noted that stimulation did not cause instantaneous movement, nor did inhibition prevent movement. Rather, both of these manipulations shifted the timing distributions. Furthermore, when we stimulated mice outside the context of the task, activation could not elicit immediate licking, nor did inhibition prevent licking, suggesting that rather than directly drive movements, dopaminergic activity influences movement timing by controlling the moment-to-moment probability of its onset, sort of like in a board game when rolling a set of dice determines whether or not you'll move on a given turn. This piqued our curiosity, and we wanted to know whether dopaminergic signals encoded the moment-to-moment probability of movement initiation in the context of self-timing. To address this question, we derived a probabilistic movement state decoding model. The general idea of this model is very simple. On any given trial, as long as the animal has not licked yet, she can be in one of two states: licking, not licking, or licking. At each moment in time after the cue, she can either stay in this no-lick state, or she can decide to transition to the licking state, thereby initiating movement. So what does this look like in our data set? Well, if we take any single trial from our GCAMP dataset and examine it in this framework, it means we can assign a lick state to each point in time between the cue and the first lick, which we can plot as a 0 for not lick and a 1 for lick. So for this example single trial, if we move forward in time from the cue, we can plot the movement state below on this state plot. Here, while the mouse has not yet licked, we see each of these time points is in the no lick state. We then assume the mouse only enters the lick state at one time on any given trial, and this happens right before we detect movement. We did this for each time point for every trial in our data set. Our task then became to decode the probability of being in the lick state for every moment in time t, given that the mouse had not moved yet. That is to say, the lick state at time t-1 was not lick. Here, just for example, I'm showing time t as this red dashed line, but keep in mind we calculated this probability for every time point in our data set. To determine whether dopaminergic signals encoded this conditional probability, we derived a nested logistic lick state decoding model. The model included the dopaminergic signal at each moment in time t, but we also wanted to incorporate the rich dynamics of the slow ramping and baseline offsets. As such, we also included the history of the dopaminergic signal before the current time point of interest. These history terms were represented as the mean dopaminergic signal within 200 millisecond windows of time, with the first history predictor representing the mean signal 1.8 to 2 seconds before the current time point, and the next predictor representing the mean signal 1.6 to 1.8 seconds before this time point, and so forth. When we then add in the beta 0 predictor, we arrive at the full model, which predicts the probability of being in the lick state at any given moment in time, provided the mouse has not licked yet. Now you might be thinking, moment-to-moment probability of movement, that seems a little abstract. Even if this model fits well, what's the ground truth for the probability of movement initiation at any given time in our data set? But it turns out this is a quantity that we can calculate directly from the movement timing distribution. To do so, we simply calculate the hazard function, the probability of licking at any time t, given the mouse has not licked yet. In other words, the hazard function is exactly the probabilistic ground truth that we want to fit using our dopaminergic signals. We thus use the results of our logistic regression to calculate a fit hazard function and then compare this to the true hazard function for each session. So here I'm showing the mean true hazard function for 12 mice calculated across 98 sessions, where each thin line represents the average for a single mouse, and the thick black line is the average across all sessions for all mice. Now, when we calculated the fit hazard function for these same 98 sessions, we see the dynamic dopaminergic signal predicts a fit hazard function that is, by eye, quite similar to that of the true hazard function. And when we overlay the fit with the true data, we better see this concordance. And it turns out that the dynamic dopaminergic signals explain 65% of the variance in the mean hazard function across sessions. So how likely was this concordance to happen by chance? As a control, we fit the same model again on the same data sets, except this time we shuffled the time points. This abolished the predictive power of the dopaminergic signal on predicting the movement state, which we can see reflected in this fit hazard function, which has no systematic structure. Overlaying the shuffled fit with the true data, we see more clearly that this model does not fit the true hazard function very well by chance, and in fact only explains about 5% of the variance in the mean hazard function. So altogether, these results show that the dynamic dopaminergic signals we observe during self-timing strongly predict the moment-to-moment probability of movement initiation. And when we take together the photometry, optogenetics, and modeling results, we conclude that dynamic dopaminergic input to the dorsal striatum probabilistically predicts and causally influences the timing of movement onset. So what does this mean in the context of the basal ganglia? Seems like what's going on is the dopaminergic signals are gradually increasing the probability of movement initiation over the course of the timed interval. This might suggest that dopaminergic signaling during self-timing does in fact work in something like a ramp to threshold process, but with a probabilistic flavor, which could be the result of dopamine's interplay with the ongoing activity of downstream striatal direct and indirect pathways. We suspect that Parkinson's patients might live in a state of low movement probability, much like the mouse when it moves relatively late, but more extreme, in which the baseline dopaminergic drive is so low that even when patients make a big effort, the system just can't ramp up very easily, requiring more time for this ramp up to occur and sometimes failing altogether, which results in delays in movement onset. To test this idea, more work is needed to understand how dopaminergic activity is relayed to and processed by downstream players of the basal ganglia, so we can really get to the bottom of what's going on in the brain that makes us move. With that, I'd like to thank the team and all you guys for your attention and to take your questions.
So much, that was great. So if anyone has any questions, you can raise your Zoom hand. So we already have Kumar, Mariana. Kumar, go ahead, please.
Thank you. That was an awesome talk. Of course, I love the ramping activity. Two questions. First question, it seems like most of your deviation from your hazard fit was early in the trial. Was that true? And the second question is, did you ever try stimulating some subset of the, you stimulated for the whole trial. Did you ever try stimulated for part of the trial, like in the middle of the trial, after the trial? Thank you.
Thank you. Okay, so I'll just revisit those questions. So the first question was whether or not the decoded hazard was most different towards the beginning of the trial, I believe. And the second question was whether we had tried stimulation at any other points during the trial time course. The answer to both of those, You know, I haven't quantitatively looked at whether or not you could say that the beginning of the trial is significantly different from the rest of the trial in terms of the fit hazard function, but I'll just go ahead and display this again so we can take a look at it. I think your intuition is correct. It does look like there's some variation beginning toward the beginning of the trial. I will just note that, I think that those reaction licks that are happening kind of right after the cue onset, those to me seem like they're potentially a different process than timing. Doesn't mean that dopamine isn't involved in those. I think that our dopamine signal suggests that there's higher dopamine at the onset of the trial when the animal reacts to the cue. But there could be something at play here toward the beginning of the trial. It just has to do with that kind of like initial reaction peak would be my intuition, but I'll have to think about that more and get back to you. The second question, did we try stimulating at other points in our trial? The answer is yes. I did try this a few different ways. One experiment was to stimulate throughout the trial course, which is the one that I ended up reporting, and that's where we got the biggest effects. The second one was I tried stimulating just at the time of the cue, just for like 500 milliseconds. I didn't spend a whole lot of time on that experiment, mostly because there were consistent results with the full-time course of stimulation, but the results were smaller. So the effect sizes were like kind of a little bit squidgier. The other experiment I did was trying to do kind of a conditional stimulation. If the animal got to 2 seconds and hadn't licked yet, I would then stimulate the animal at that point. The results from that were very interesting, and I'd love to talk more offline if you're interested in this in particular. But there was kind of like a sigmoidal pattern. There was kind of an immediate delay almost right after you stimulated, maybe like the animal was startled a little bit. And then rapidly, you kind of got onset of movement after that. I was kind of expecting, based on the full-time course stimulation, that you would trigger like an immediate movement, or you'd be closer to triggering a movement. I think there's a little bit of a confound here in that, I think if the animals can sense the light coming on, there is a little bit of a startle effect. I will say that in our control animals where we did stimulation, there was no consistent effect of light stimulation, but whatever effect like that we saw, even though it wasn't significant, it did seem like they were a little startled by the blue light. So another way to get at that might be to try with like crimson R, for example, for your stimulation. I hope that answers those questions. And again, happy to talk more. Let's see.
Please go ahead.
Is that me? Okay. I think you might have answered this partly, but you introduced this by referring to this as self-initiated movements. And I'm wondering if you gave a reaction time task, if you'd get similar neural data.
You mean just like reacting to a simple sensory stimulus and then looking
Yes.
at dopamine signals to that? I will say that although we did not do that exact task, I think you can kind of make some inferences about that based on the task that we did do when the animals do react to the cue. I'll just tell you what I think my intuition is about this. What we did observe is that, let's see if I can actually go back to this. Yeah, it might be too far up. I'll talk while I scroll so we can make our time efficient. For those very early lick times, you see that you have this like higher baseline. Now here I'm not showing any of the reaction licks, but what you kind of see on those is they start from an even higher baseline level. So you kind of have this like high dopamine signal coming into the trial, cue comes, and then they just kind of lick. But those signals layer on top of here in a nice rainbow as well. You can kind of keep the rainbow going upwards if you look at some of those earlier movement times, which we didn't focus on. What do I think that means? I think it could mean that the system is already kind of predisposed to move and you get that sensory cue on top of it. It could be just like injecting more momentum, And I think that our overall view altogether is that, dopamine is not acting alone. It's not like a motor neuron, right? It's kind of participating with downstream players. It would make sense to me that other systems could kind of speed up your reaction, or rather having that high dopamine tone going into a trial could speed up your reaction time. I think that is consistent with what we see. It would be interesting to repeat that task. And I think other people probably have on that. Yeah.
Sorry, just to clarify this. So in a reaction time task, you wouldn't expect variability at 0, like what you're showing here.
Oh no, I think you would. But
okay.
Here's the thing. With the reaction, this is one of the nice things about the timing task. If you look at the timing task, we're looking at pretty like broad ranges of time where we can really separate out the signals well. Suppose that I had done like a 500 millisecond timing task instead, right? Now I'd just be looking at, well, let's pick a different interval. Let's pick like 0.75 to 1, these two traces here. Now my difference in baseline between these two traces, it's super similar, right? It'd be very, very difficult to tease those two apart. And I think what you would find if you just simply did a reaction task is you might be able to find some correlation in baseline level or post-cue, stimulus dopaminergic activity, but it might be really small. It might just be hard to tease that apart. And I've suspected for a long time that maybe the reason we've been able to see these kind of baseline effects is because, first of all, we had the inkling to look at variation in time, right? Because it's a timing task, but also because we had a large amount of variability to start with. It just kind of like lends itself nicely to this kind of analysis. Whereas I think other tasks that only look at one movement time point, maybe they don't do that, as easily or as intuitively.
Okay, thanks.
Thank you. Good question.
Peter Balsam.
That was a great talk, great work. Really nice to see it. I want to ask the question that everybody in the room has, which is there, because you're looking at a bulk signal, is there anything in the data that makes you think that it's ramping neurons or the recruitment of a population?
Oh my gosh, I love this question and I want to answer it. As you know, I can't answer it, but I can tell you my thoughts on it. I think there is a simple way to go about this. We've got these great these endoscopes that you can really look at actual individual neural signaling using these calcium indicators. Our lab is not a very rich lab. We didn't have those. They're apparently very expensive. So I think that experiment is doable. It's just something that we unfortunately we're not able to do due to our limitations and resources. But what do I think? I don't know. I think something that's very interesting about this signal is if you compare these, if you look in the paper at figure 2 supplement 2 or something like that, you'll see the actual reward related responses because we plot past the actual movement time and we look at the rewarded trials, the unrewarded trials. The reward related responses are so much larger than these timing related responses. I mean, it's like 10 times larger, right? Suggests that there's something getting recruited in to produce that reward related response. Does that mean that like a subset of the neurons are slowly increasing their activity? Does it mean a subset of the neurons are kind of recruiting their nearest neighbors slowly during the timing process? I think there's reasonable hypotheses across the board about that, but I think it does suggest it's a different process potentially than what I assume is going to be like some big burst responses happening with reward, which I assume recruits a large population of dopamine neurons, but I can't answer that question. I wish I could. Yeah. If anyone else wants to try, let me know. If you have money, I'll be happy to try with you.
Okay, we have one last question in the chat. And iPad Mini asks, do the dopamine peaks and the ramp occur in highly overlapping or segregated regions in striatum?
That's a cool question. All right, so I only recorded from basically one area of striatum. The only evidence that I can give you or an answer that I can give you is based on an experiment where accidentally I broke the fiber in one of the mice and I couldn't implant the dorsolateral striatum. So I had to plant dorsal medial striatum. And that mouse, I didn't see a whole lot of ramping stuff going on in that target. Does that give you any information whatsoever? I'm not sure. All I can say for now is simply that where we looked in striatum was a consistent location. We kind of saw that you had these little ramps that went up in that location, and they looked very similar to the signals that we saw at the cell bodies. Based on what we know about dopamine neurons, they project pretty diffusely. It's, I think, reasonable to assume that that signal is getting broadcast to a lot of different places. And I don't personally think the specificity in the animal's movement comes from dopamine neurons. In fact, I think there's good reason to suggest it doesn't. For example, if you just whack dopamine neurons really hard with optogenetics, you can get them to run around. You can get mice to run around, but you can't get them to do a precise movement like licking. And in fact, we tried that experiment outside the context of the task. We couldn't get the animals to lick, right? Suggests that what dopamine is doing is it's tuning the onset of something that downstream system, the striatum, cortical striatal system, those loops have already kind of planned to do. And that's what I think is going on. So I suspect the signal is probably broadcast pretty diffusely, but is maybe, in concert with that other signal, which is more specific, maybe from cortex, that's decided on the movement that wants to happen. What evidence do I have of that? I don't have a whole lot. I will say, there is one last thing, and I want to make sure I give time to Francois. We did do the tapered fiber experiment where we did record a bulk signal. we don't know where that signal is coming from. It could be like anywhere along that tapered fiber shank. But I think a very interesting experiment to follow up would be to use, they have these new like kind of motor, like servo motors that can move the recording site up and down the fiber optic shank in those tapered fiber optics. That's something we're very interested in exploring. And obviously this is another question that's amenable to things like endoscopes if you have lots of dollars. I hope that sort of answers that question.
Awesome. Thank you so much, Allison. This was great. I think we can move on to the second speaker.
Thank you.
All right. Yeah, thank you. All right. We have Professor Foerster, and he will talk on the neurocognitive mechanisms of enhanced implicit temporal processing in action video game players. So please go ahead.
Thank you very much. So you hear me well? Okay. So thank you for having me. I'm very honored, and as it's the first time I'm invited to this time research forum. And so first of all, I wasn't sure about this audience. So I don't know if most of you are neuroscientists or you come from more psychology or neuroscience. So I try to be quite not a bit broad and to be sure I'm understood by everyone. So I hope I will not bore some of you. Okay, so I'm François Foerster. I'm a postdoctoral fellow in France at the University Hospital of Strasbourg, and I'm part of the Angers lab. And so we work in psychiatry. So I'm doing cognitive neuroscience about and studying the temporal processing in different populations, healthy volunteers, but also psychiatric patients, especially schizophrenia patients. And now I'm going to talk about the recent work I've done with people that play video games and their ability to implicitly process time. So just trying to fix this bar. Okay, so animals and humans, we need to adapt constantly in this changing environment. And the best way, one of the best way to do it is to anticipate all our sensory signals in space, but also in time. So beyond the fight or flight situation of animals, us humans, we have these timing mechanisms that are very important in, for instance, artistic activity, dancing, music, that has been largely studied. Also in sport, of course, as some of you might have seen, like the Olympic Games, when the motor timing is very, very crucial to outperform to our task. And we were interested in our European Consortium of Research in Psychiatry about video games. So basically in many, many video games, as you can see here on the right side, you have this kind of very typical situation when basically, again, it's about motor timing. You perceive some information very quickly and you need to behave in a very specific way at the exact right timing. And because you have this exposure thousands and thousands of times in this population, the players that plays hours for every day for years and years, our hypothesis is that all this implicit learning of time intervals can be completely automatized. And so maybe the ability to implicitly process time is altered. So, so could video gameplay influence our implicit time processing? And this is very important for psychiatry because there's a new trend in the field that proposes that actually video games could be one of the best tools we have for cognitive rehabilitation. And the idea is simply that video games are highly engaging. And so they could be a tool that actually, a solution that where patient could actually practice this. So what we want to do, some of us in psychiatry wants to do, to design proper rehabilitation tool that are actually engaging people so they would actually use it. And so to study implicit timing, we use the variable paired task. So I think that some of you might know, as given the talk before. And so the simple idea of the variable paired task is that you have this arrow of time, you have a point t in time, your present moment, and you have the passage of time. And if you have to anticipate something in time, you will, for instance, anticipate something that close at the near future, time plus 1 and time plus 2. And if now you have sensory signal such as a visual cue like this, a red light, you will anticipate that the state of the world will change. So this red light will become green at some point. And so as time elapses, the chance for this light to change state, become green, increases. So this is reflected in the work we saw before, of course, the other function. And we use this task, the variability task, to study implicit time processing. And so in the lab, we simplify these like before, with, for instance, two simple time intervals, time plus 1 and time plus 2, 400 milliseconds and 1 second. And so here, basically in this situation, you only have the passage of time that helps you to anticipate the stimulus in time. But there's another situation when you have explicit information that tells you, that can guide your temporal expectations. So for instance, if before this signal, I tell you in 400 milliseconds, so time plus 1, you have the target. In that case, when you have the signal, you will have temporal attention mechanisms that will allow you to know that 100% of the case, the stimulus will appear at that moment. So these are the targets, so I can prepare in a specific way my motor response. And so this temporal attention is important because it helps, it allows to mobilize our cognitive resources when is it required, so we can use better strategies to manipulate our resources. And from the data, we saw that, of course, that optimize our behaviors in the sense that our responses are faster or more precise. Okay, so what we did, we used this task in people that play video games and controls, of course, and we use this task in virtual reality. So the point of using virtual reality, again, is because at the end of the day, what we want is a tool that could be used in clinical settings. So doing a task that is not just a classical experimental psychology task that can be quite boring sometimes, but finding some way that is very engaging for the participants. So not only for the participants that come back to the lab, but at the end, if we found some markers that are very interesting, we could actually use it in clinic. And so we have these people that went in the lab, they experiment in virtual reality, and the task is last presented here. So we have first a green light that tells us that the target will come at some point. It changes state, it becomes gray. And when it's gray, there is time plus 1 or time plus 2, these two for period, these two time intervals. And then the target appears, the red light, and they have to press the button as fast as possible. So I have a video that shows that. We just try to move slightly like this. I hope everyone will see it quite correctly, because with internet, we never know. So people are in virtual reality. They have this controller in their right hand. They have the different robots. There's a green light. They look at it, turns gray. This is the waiting period. Then they have a target, and they press it as fast as possible. So the idea of the experiment still is very basic, even though it's virtual reality. It can be still, unfortunately, a bit boring. And so here, as you can see, there is two types of robots. They were giving contextual cues. They are colored and the gray robots. And the idea here is that in one case, people have to play with this gray robot. That's what we call the neutral cue condition. So people have no idea when the target will appear, either time plus 1 or time plus 2. And in the other case, they play with this colored robot that plays the role of temporal attention. And they tell them in advance if the target will appear at time plus 1 or time plus 2. So in the first case, they have to anticipate in time the target only based on the passage of time, which is completely implicit because you don't have to think about time. It's highly automatic. And in the other case, you use a temporal cue, which is explicit. It's an explicit information. And so you have temporal attention mechanisms that will help you to anticipate and prepare your motor action. Okay. And so our population were the people that play video games, action video games to be precise. And these people were playing basically on a daily basis. Usually it's like more than 20 hours a week. So it's quite intense. And control population, a sample, of course. So people that don't play video games that we call the NVGPs, non-video game players. Okay. So the first thing we're interested, of course, is the reaction time, the behaviors. And so what we expect in this traditional variable paired task is this kind of patterns. So people would be faster to respond to the target at the second interval than the first one. Not only because they have more time to prepare the action, but also because at that moment, at the time interval, the T+1, they are not sure 100% that the target will appear at that moment. Whereas here, 10 plus 2 is the final interval. They know 100% it will be the case. So what we're interested, of course, is this slope, the difference between these two periods. And this slope represents somehow this implicit benefits from the passage of time. So much people benefit from the passage of time. And what we're interested in this task, of course, is this slope. But I propose a specific calculation that is, for instance, that is called the gain in other psychophysical task. So we're interested not only in the difference in milliseconds, but for each participant, so independently of the average speed of response, what is the speed change? So how much faster people are here, time plus 2, than compared to the situation in time plus 1. So it's very simple formula. We take a baseline condition, for instance, condition A here in green, minus condition B, we divide by condition A, and we multiply everything by 100, so to have a percentage. And if we change these variables with our values, so here in that situation, that person will be 20% faster at time plus 2 than time plus 1. So the benefit from the passage of time will be approximately 20%. And that's interesting. The interest in that as well is that we reduce the variability between participants. And so we apply this formula in gamers and non-gamers. And of course, in the case of the neutral Q, so it's only based on implicit processing of time. There's no attention. In the case, there is attention. So with the temporal Q. And here is what we found. So on the left side, you have the gamers. On the right side, you have the non-gamers. And in black here, you have the benefit from the passage of time, this calculation in the neutral Q condition, and in red in temporal Q condition. And so what we found is basically a group difference on the neutral Q, such as gamers, actually, they benefit better from the passage of time compared to the controls. And this is only in the neutral Q condition, not in a temporal Q condition. So that goes in favor of our hypothesis that maybe they have better implicit temporal processing, these people that play video games on a daily basis. And we use the same formula to see if people, how much faster people would be using the Q. So we just replace these variables with here temporal condition versus neutral condition. And what we found is that basically everyone benefits from the temporal Q. That's why the values are mainly positive, but there's no difference in the groups. So people don't, these gamers don't benefit more than controls in using the temporal attention. So now from the data, we try to understand what are the mechanisms behind that, how can we explain that gamers play benefits better from this passage of time. And what we did is during this experiment under this Vtr8 headset, people had wear an EEG. So we have EEG recording during these sessions. And the first thing we look at the CNV. So actually, I think most of you might know. And we didn't find group effects in these. So I skipped that part. And but what we looked at is also two different markers of temporal expectations. And we based our work from André Cravo and a paper has been published 10 years ago. So the first thing is the theta band recorded in frontal lobe. So in the paper, they show that the adjustments in theta power contributes to a central mechanism for controlling neural acceptability according to temporal expectation. So when we expect in time a percept, a stimulus, your theta power will be modulated. So we looked first if we have this theta power in a task, and indeed, this is what you can see in red. So the red area is basically the increase of theta power during the weighting of the target. So we have indeed this oscillation present in our data. And then we looked at it in the two groups and in the two conditions. So in the neutral Q condition, these gray robots, here, basically you have the warning signal and the targets over here appear. And we're interested to compare analysis like with the original paper. So we looked at the data over here, which is time plus 1 when the target could have appeared but did not. So basically there was 50% chance to have the target over here. And here is the temporal Q condition. So it's similar, except that in advance, people know the target appears over here. So here, technically, they know there is 0% chance that the target appear. So it's either 50% chance or 100% or 0% chance. So looking at this data, we found two main effects. One main effect of the group, because gamers have lower theta power than controls, and the effect of the Q, such as in the neutral Q condition, the theta power is increased compared to the temporal Q condition. And so from these data, we draw two conclusions. The first is that this theta power is increased when the target probability occurrence is higher, which is here in the neutral Q condition, 50% chance. So that goes with the paper in the same direction, that there's a link between temporal expectation and mid-frontal theta power. And that helps us to understand what happens in gamers, because here in gamers, they have a decreased theta power in red. And it's possible they have decreased temporal expectation compared to controls when the probability of target occurrence is indeed low, because you see the 51 chance over 2 or 0%. So is it possible that they have better adaptation? The strategies to apply the temporal expectation might be better adapted in gamers. And so we follow up this analysis with another one, as in the papers, that show that maybe it's related to specific neural communication strategies. So what we did is this phase power coupling in theta and beta band. So I will just try to explain clearly how this works. This is what we call the functional coupling in EEG. Imagine two types of oscillation. One that is very slow, like here in blue. This is, for instance, our theta power that we found in the mid-frontal area. And there's another oscillation that is faster, here in green, that we can find in the frontal cortex as well and in the left motor areas. And just to remember, in this task, participants answered with their right hands. So that's why probably we have a beta on the left motor area. And so the idea is that if we have a perfect coupling, so this is just an example, it means that when you have a specific phase value of this theta power, you have also a response of another oscillation in another part of the brain. And so here, for instance, every time the oscillation is a specific position, we have a response in other brain areas. So there is a functional coupling that is at work somehow. So we look at this coupling between this frontal and left motor area in the two conditions and in the two groups. And so what we found is here in gamers. So basically here, to be short and simple, the more yellow it is, more coupling there is. And if it's completely black, there is no coupling at all, which is almost impossible. And so what we found is that there is a stronger coupling in the neutral Q condition compared to the temporal Q condition in gamers. So when there's no attention, we have this coupling. And when we compare with controls, there were no difference. So maybe this functional coupling is involved indeed in the ability to implicitly process time. And so there might be a relationship between this coupling and these benefits from the passage of time that I proposed before from the reaction time data. And this is what we found indeed. So basically, the higher is the coupling, more people will benefit from the passage of time. This is what we see. And this is from the data in the neutral Q condition when you don't have attention. But when you have the temporal attention, also this coupling disappear. I mean, this relationship between the coupling and the benefit from the passage of time disappear. So maybe indeed gamers, they have a specific communication between different brain areas that can help them to better process time. All right. And the last point I wanted to talk about, I think I have, it's a bit tight on time, but I will be quick. Because now more and more labs try to create to set up Vtr8 experiments for many different reasons. And for this experiment, we use Vtr8 as embedded night tracker. So you can actually record the ocular movements during the task. And so this is very great for cognitive neuroscience and psychology. And so we're interested in one thing in particular. If I ask you to look at this red dot in the front of you, you will think that your gaze is completely stable, where actually it's not. If we look at the eye tracking data, we would find tiny saccades. That's what we call microsaccades, and we are not aware of these. What is interesting is that it has been found the last few years is that these microsaccades are basically inhibited by temporal attention. So when you know that a stimulus will appear, you will have the probability of doing one of these microsaccades will be lower, inhibited. So when we look at our data, here, this is the probability of doing a saccade over time. You have the first signal, then you wait, you have this response. And then here, you wait for the target that appears at that moment. And indeed, the probability of doing a saccade is way lower in the temporal Q condition. So when you know 100% of the time the target appears at that moment, than when you do not. And what we were interested here, however, concerning our gamers is this response. So when you have to process this first signal that tells you, okay, you have to wait and anticipate for the target, you have the probability of doing this arcade is very particular. There is an inhibition and then a rebound. If you look at the data, recent data that has been collected with traditional eye tracking system, it's very similar. And the idea is that here, this response reflects somehow the processing of the stimulus, and it can be modulated, for instance, by attention. This was for this task from the paper cited. And so we looked at this response to the first stimulus in gamers and non-gamers. And what was striking is that basically the response is the same, except that it just occurs earlier in gamers. And to do so, we calculated the latency of this peak inhibition and the latency of this peak rebound, and both were shorter in gamers than non-gamers. So maybe this can explain as well why they benefit better from the passage of time and the relationship with their implicit time processing. And actually, what we found using another correlation is that people that benefit the most from the passage of time from the index we calculated are the people that have the shortest oculomotor response. Okay, so to wrap it up, because maybe I'm short on time, just a few take-home message. So from marketing reports published last year, we know that more than 70% of the population between 6 and 40 years old play video games. Can be at home or in the public transport, anywhere. And the average rate is 7 hours a week. So it's huge and it's increasing. So we have a huge part of the population that practice these video games. So it's very important to understand what are the benefits and the costs of this practice. And especially if there is benefits, we need to take them and maybe it can influence what we can do with other people for cognitive enhancements and rehabilitate some patients, for instance. So from this data, we found that an enhanced implicit temporal processing in gamers. So maybe we could use these video games as a base to design intervention and program for rehabilitation with people that have deficits in temporal processing, such as schizophrenia or bipolar disorders. We found that action video games might shape some neural mechanisms that are involved in temporal cognition. We need to understand what are these. We have some ideas, but we need more research to replicate these results. And it seems like action video games can affect our oculomotor systems. Why and how is still very obscure. We don't know, but there might be a relationship between oculomotor response and temporal processing. And I will thank you for your attention. I thank you, my lab, the INSERM in Strasbourg, and thank you for your attention.
This was a good, interesting talk. So if anyone has any questions, please go ahead and raise your Zoom hand or type in the chat. Question, were the reaction times generally faster for gamers?
Okay, yeah, thank you, sorry. So indeed, they were slightly faster, not as much as expected, but they were slightly faster. But what was important is basically we had a lot of different variability in the two groups. A huge variability in the non-gamers, but in the gamers, they were all the same profile of reaction time. And so we had to face a problem of variability in the two groups. And that's why I came up with this speed change index. So somehow it completely reduced all the variability between people. I think it replies to the question. I see another question from Kevin. This temporal processing advantage is in gamers is a few milliseconds. How much do you think this transfers to other abilities? How big is the effect in athletes? It seems crucial for the rest of us. How much of an impact does it have on cognitive abilities? So indeed, it was a few milliseconds, but that's why actually this index actually it's important because it's not just about a few milliseconds, but you have to think a few milliseconds compared to what is the basis. So here, indeed, it was not too much in this task. It was like between 5 and 10%. And how much it transferred to other abilities and how big is the effect? I don't know yet, really, because this is very new. So these data are like a few months old, and we started replicated this result and look at them in patients, for instance, in psychiatry. So I don't know much yet. And in athletes, indeed, it's crucial. And for the rest of us, I don't know, for the cognitive ability, maybe really depends on the task, really. So I would be interested to know how this happens with this index, for instance, in musicians or dancers or other athletes, indeed. But I don't know yet.
Thank you. And Well, yeah, I mean, actually, I believe that probably has a big impact because we're having lots of little decisions all the time. And if all of these are improved by a few milliseconds, then a whole thought process might improve by, 100 milliseconds or something if each time it's a small bit. But I thought the question was interesting. So thank you very much.
Indeed, you're right. So it really depends. If you think in a situation of multitasking, this can be very important indeed. But I have no idea. I don't study multitasking, but indeed, it's a very good idea. Actually, very good point. Thank you.
One last question, Kumar, go ahead, please.
All right, two quick questions. Your talk was beautiful. I was wondering if the theta was locked to the Q and if you'd ever looked at errors.
Okay, so just to be sure about the errors, indeed, I didn't present them. And what we did the errors, so basically when people press the button before the target, these are the errors you're talking about, anticipation. And so, yeah, indeed, we look at this anticipation, and gamers, these people that play video games, do less errors. So we can think that about motor impulsivity. Actually, they're reduced compared to what people would think in the literature, saying that games, playing games make you impulsive. Actually, we found the opposite. And so yeah, please.
I'll bet you that the errors trigger a prominent theta response that lasts. So it might be interesting to look at.
Indeed, I didn't look at the data. Actually, I get rid of this data because you have a motor response during the waiting. So it's a problem for EEG analysis for clean data. But actually, this is another type of data that will be interesting to look at. There were very few, like 2 to 5% of the time, because fortunately, people did the task we asked them to do. So what are your thoughts on the application of this project on rehabilitation? So there's different things. And first of all, actually, I'm writing for, I'm trying to get a grant for a proper rehabilitation project here to do this really an intervention and see really if video games change this, create this effect, to be sure. And so the idea is really, if we know that, for instance, at least in schizophrenia, there is disorders of implicit time processing, not explicit. So somehow there's research that shows that this implicit time processing could be related to the cerebellum. And so the idea of practicing something like video games, doesn't have to be video games, something like it, similar, that really train these mechanisms, somehow some patient could really benefit from it. But there's no data on that. No one did ever anything like this. And I hope the European Commission will give me the opportunity to show that if it does or not. And rehabilitation. So the thing is, they remind me that in my data, I don't know, but I know that the FDA in the US, they accepted a kind of video game like this when kids from 8 to 12, if I remember correctly, 8 to 12, they play these kind of specific video games for ADHD. And the paper has been published in The Lancet, and it's really great results. And so that's why now there is actually a lot of grant and research that is done to know if we can extend to other pathologies, psychopathologies. And that's why in my consortium, we're interested in schizophrenia, bipolar disorders, depression, autism, and yeah. So it's great avenues, I think. The idea of video games, we have to think that really the idea is not to use video games, but to inspire designers and programmers to design really tools that could be used for clinicians, which is something that for clinicians, it's another part of the world. But it could be very great and helpful for people.
And just one point about that: the control groups are important too. In that FDA-approved video game, the control group was, I think, a puzzle-solving game, which wasn't exactly the same kind of comparison. So I mean, it's great that it got that, but in general, in designing these, I would recommend to make very important, careful decisions about the control groups to improve the
absolutely
validity.
Yeah, to be sure that it's actually the specific aspect of your activity is not just any activity that is close to just being stimulated. Because of course, if you compare that to just doing nothing, a lot of things that helps compared to doing nothing. All right.
Do you have another question or?
No, sorry, I'll take it
No
down.
worries. Of course. Okay. All right. Thank you so much, Francois and Allison. These were really great talks. So I just wanted to end by saying that our next talk will be by Dr. Mark Howard from Boston University, and it will be on March 15th. We will send out the announcement pretty soon. So thank you all so much again, and hope to see you all soon. Bye.
Thank you very much.
Thank you.